Good evening everyone. Welcome to another free session specifically for your GATE XL examination, right? You all have your examination on 7th of February. So it's less than a month left. So let everyone join in and then we'll start with the class, right? So it is the 4th session in our question series. So every single day we are solving out questions. We are practicing as many questions as possible so that you can revise the question, like you can practice out as many questions as possible, right? So just confirm whether the audio and video is clear to you or not. If yes, we'll begin with the class. Yes. Just check whether the audio and video is clear to you or not, then we'll begin with the class accordingly. Many of you are there in the class already. Some lighting thing today. Perfect. Let's begin. So just a quick reminder, our CSIR net rankers batch is ongoing and we are preparing in the best manner possible for the upcoming June exam. So if you're someone who's preparing for June attempt, enroll in the rankers batch, which is built specifically for results and not for you to get distracted and get confused. Then we have for the GATE examination, we have the GATE ebooks available, GATE revision capsule available and the test series as well. So whatever is your requirement, you can have it. You can WhatsApp us on this number for any discussion. Our rankers batch is ongoing. You can join our free Lakshya batch as well. First question, which of the following carbohydrates have or has a beta 1,4 glycosidic linkage? Mark the answer. Which of the following carbohydrates have beta 1,4 glycosidic linkage? Cellulose, chitin, lactose, maltose. What should be the answer? Which of the following carbohydrates have beta 1,4 glycosidic linkage? Easy. Mark the answer. Just give me 1 minute. Let me turn on some more lights. Okay. Mark the answer for this particular question, everyone. Which of the following carbohydrates has or have beta 1,4 glycosidic linkage? So what should be the answer? So cellulose has glucose linked in beta 1,4 glycosidic. Chitin has beta 1,4. Maltose has beta 1,4. Your lactose has beta 1,4, but your maltose have alpha 1,4 beta link, alpha 1,4 linkage, right? So what should be the answer? The answer should be, because this is a MSq multiple select question is this, right? So cellulose, chitin and lactose, all three are the correct answer. A, B and C, all three of them have beta 1,4 linkage. Now similar concept question can be asked in the format of which of the following does not have it, right? Then the answer would have been maltose, right? So we have to make sure that we are looking into the answers according to what the question is asking us. So over here, has or have, this means it is asking you to find one or more than one, like it could be one or more than one options being correct, because we know in your GATE exam you have more than like different type of questions and one of the format is multiple select questions, right? So make sure that you remember this. Another question for you all, which of the following statement about IgA is or are correct? When we learn about IgA, that is immunoglobulin A. We have the family, 5 of the family members of immunoglobulins. So IgA it is talking about. It is secreted into colostrum. It is transported across the cell by the transcytosis. Its secretion is facilitated by poly Ig receptor, and it primarily exists as a dimer in the serum. Mark the answer. What should be the answer for this question? Which of the following statements about IgA is or are correct? So you can have one or more than one statement being the correct one. So they are secreted in colostrum, they are transported across the membrane, across the cell by transcytosis. Its secretion is facilitated by poly Ig receptor, and it primarily exists as a dimer in the serum. Very good. Let's see. So when we talk about my IgA, immunoglobulin A, so it is secreted in colostrum. It crosses the epithelia by transcytosis via a poly Ig receptor. So A, B, C, all of these three are correct. Fourth is, in the serum IgA, it is mainly monomeric. Dimeric in secretion is the wrong statement. So A, B, C. A, B, C. We need to find out the correct statement. Similar concept could be asked about the incorrect as well. It might give you same option and it can ask you which of the following is the wrong one. So what should be the answer? Wrong should be option number D, because it exists as a monomer, right? So keep a note of this thing as well. Hope it's clear. Next question. Which of the following enzymes converts your testosterone into estradiol? Yes, mark the answer for this. Which of the following enzyme converts testosterone into estradiol? Aromatase, 3-beta dihydroxysteroid dehydrogenase, 5-alpha reductase, 17-beta dihydroxysteroid dehydrogenase. What should be the answer? Mark this. Yes. Try to mark the answer for this. Which of the following enzymes converts testosterone into estradiol? Factual statement. Okay. Which of the enzyme converts the testosterone into estradiol? Very simple. So it is your aromatase which converts the same, right, which converts your testosterone into estradiol. This is a normal MCQ question, okay, no other MSQ or any other thing. Next question. Purification of 6 histidine tagged protein using your NINTA column is an example of which of the following chromatography? So when we learn about the different type of chromatography, one of the segment in one type of chromatography is purifying it with the help of histidine tagged proteins, right? So which of the following, in which chromatography do you get to see that? Mark the answer for this. Yes, what should be the answer for this particular segment? Mark it. Let's see. So, with the help of or purifying the histidine tagged protein is found in which of the following chromatography? So, it is your affinity chromatography, right? So, in this a specific ligand protein interaction is taken into consideration and the final work is completed, right? So, affinity chromatography is the answer for the same. Okay, next question. Okay, try this question everyone. Which of the following molecules do not absorb at 280 nanometers in an aqueous solution of pH 7 at room temperature? Which of the following molecules do not absorb at 280 nanometers in an aqueous solution of pH 7 at room temperature? Polydeoxyguanylate, adenosine triphosphate, phenylalanine and tyrosine. Which of the following molecule does not absorb at 280 nanometers? Try this. Okay, very good. Which molecule does not absorb at 280 nanometers? So, adenosine triphosphate. Okay, so 280 nanometer absorption mainly occurs from your aromatic residues or aromatic nucleotides that we have. So, ATP absorb at around 260, not 280. All the other three absorb at or nearly around your 280 only, right? So, what should be the answer? B. Very good, very good. So, adenosine triphosphate is the correct answer for this particular question. One more question. A molecule that forms a donor acceptor energy transfer pair with dinesyl group is. So, which molecules forms a pair of donor acceptor energy transfer pair? Mark the answer for this. Give it a try to this question. A molecule that forms a donor acceptor energy transfer pair with the dinesyl group is. Quickly mark it, then we'll do it together. Mark it everyone. Okay, so the molecule that forms a donor acceptor energy transfer the pair with the dinesyl group is aspartate, histidine, lysine or tryptophan. What should be the answer? Your tryptophan. So, very good, very good. D is the answer. D, tryptophan. It's a strong donor of in the feet with dinesyl, so it acts as a energy acceptor. Tryptophan is the answer. It is the tryptophan, not histidine. D is the answer for the Question. Next question. The stationary phase that is used in the gel filtration chromatography is composed of which of the following? Last time homework answer is 3 ATP. Yes, 3 ATP was the answer. The question was for consumption or for fixation of, I think, 1 carbon dioxide, how many ATP will be utilized in 1 Kelvin cycle, something like that the question was, right? Very good. Why didn't you join in the yesterday's session? Yes. The stationary phase used in the gel filtration chromatography is composed of which of the following? Blue dexin, carboxymethyl cellulose, diethyl aminoethyl glucose and seprose. Mark the answer for this question now. Stationary phase used in the gel filtration chromatography is composed of which of the following? The stationary phase that is used in the gel filtration chromatography is composed of which of the following? Blue dextrin, carboxymethyl cellulose, DEAE sephirose. Quickly mark it. Let's see. It is option number D. So, gel filtration, which is your size exclusion chromatography stationary phase, it is porous beads like Sephirox, right? Blue dextrin is void volume marker, not in the stationary phase. Then the CM cellulose, it is a cation exchanger, and DEAE cellulose is an ion exchanger, right? So, my stationary phase is usually composed of option number D. Keep a note of this thing. All these are 2 marker, 1 marker, all clubbed in together. You can get any question asked in the examination again in any other format. Maybe a match the following, maybe a multiple select question format, maybe a multiple choice question format. So, any of the format can be asked in. Next question, very simple question. Now, this is related to your wobble hypothesis. So, according to the wobble hypothesis, inosine at the 3rd position of anticodon cannot form the hydrogen bond with which of the following? So, when we learn about the wobble hypothesis in unit number 3, that is molecular biology, so it is asking us that inosine, which is at the 3rd position of the anticodon, it cannot form the hydrogen bond with which of the following? Adenine, cytidine, guanine or uracil? Very simple. Direct statement question from wobble hypothesis. Mark it. Mark it. In Wobble hypothesis, the inosine which is present at the third position cannot form a bond with which of the following? So it cannot form a bond with guanine. It cannot pair with guanine. Now you haven't learned the facts properly, right? It can form the bonds with your adenine, cytidine or uracil in the codon very easily, but with guanine it cannot make up the bond. So C is the correct answer for the question. D, very good, D is the answer. Okay, now another question. Now this one is a statement, or I would say factual question. So pK value you need to find out of arginine in your gonidium group. In the gonidium group, what is the pKa value of arginine? Mark it. Mark the answer. This one is a factual statement. What is the pKa value of gonidium group of arginine? Done? Quickly mark it. Okay, let's see. It is 12.50. Very good. So gonidium group in the arginine side chain is, yes, it is 12.50 only. So the gonidium group of the arginine side chain is strongly basic and it remains protonated at physiological pH, right? So it has the approximate value of 12.50, right? So D is the answer for the same. Next, the non-coenzyme vitamin is, which of the following is a non-coenzyme vitamin? Mark the answer for this. Non-coenzyme vitamin. Done? Non-coenzyme vitamin. Let's see. It is your ascorbic acid, which is a non-coenzyme, right? So question is, which of the following is a non-coenzyme vitamin? So it is your ascorbic acid, that is vitamin C. It functions mainly as an antioxidant and in hydroxylation reaction, but it is not a coenzyme. So folic acid, nucleic acid and thiamine, they are all precursors of the coenzymes of different segments like THF or NAD plus, TPP respectively, right? So what should be the answer? So my non coenzyme vitamin is only ascorbic acid. All other three can work as a coenzyme in one format or the other, right? Hope it's clear. Another question, telomerase has a function similar to which of the following? Good evening. Good evening, Vasudev Joshi. Mark the answer for this. Telomerase has a function similar to which of the following? DNA dependent DNA polymerase, RNA polymerase, DNA gyrase, reverse transcriptase. Mark it. Very good, very good. Telomerase has a function similar to reverse transcriptase, DNA gyrase, RNA polymerase, DNA dependent DNA polymerase. Very good. Its main functionality is similar to reverse transcriptase. So telomerase is what? Telomerase is a ribonucleoprotein enzyme that carries its own RNA template, right? It extends the 3' end of the linear chromosome by synthesizing the DNA from the RNA template. Same functionality is performed with your RNA transcriptase as well. So what should be the answer? Answer should be option number D, that is your reverse transcriptase. It has a similar functionality of reverse transcriptase. Next question, which of the following enzyme is used in polymerase chain reaction? Which enzyme is used in the polymerase chain reaction, or we call it PCR? Clinau fragment, Taq polymerase, primase, T7 polymerase. What should be the answer? Which enzyme is used in RNA in the polymerase chain reaction? Mark the answer. Clinau fragment, Taq polymerase, T7 polymerase, primase. Okay, okay, very good, very good, very good. Let's see. Yes, it is your Taq polymerase. So the PCR requires a thermostable DNA polymerase because the reaction involves repeated heating to 95 degrees Celsius for the DNA denaturation. Taq polymerase is heat stable and was first widely used in the PCR. Then Clinau fragment is used in the DNA sequencing and labeling. T7 polymerase is a phage RNA polymerase that is used in the in vitro transcription. Then primase synthase RNA primer in vivo, not used in the PCR. So correct answer is Taq polymerase. Next question, in hepatocytes the detoxification of drug occurs in? Yes, next question. In your hepatocytes the detoxification of drugs occur in Golgi apparatus, nucleolus, rough endoplasmic reticulum and smooth endoplasmic reticulum. Mark this. Mark the answer. hepatocytes, the detoxification of drug occurs in the smooth endoplasmic reticulum. Obviously, very basic information or a very basic question altogether. So, smooth endoplasmic reticulum is a site for cytochrome 450 enzyme that oxidizes and detoxifies the drugs, toxins and xenobiotics, right. Then you have the Golgi apparatus for the protein modification. Nucleolus is for the one that makes up the rRNA, and rough endoplasmic reticulum is for the protein synthesis. So, question was the detoxification part. So, detoxification will be for detoxifying the drugs, toxins and all the other segments. Another question for you all. Okay, which of the following cells lack HGPRT? So, when we study about this segment, there's a very this is the very initial segment or very initial information that we learn. So, which of the following cells lack your hypoxanthine guanine phosphoribosyl transferase, HGPRT? B cell, T cell, macrophage, myeloma. Mark it quickly. Which of the following cells lack hypoxanthine guanine phosphoribosyl transferase, HGPRT? Done? I don't know. Okay, no worries. We'll see. It's your T cells that lack the same. Now, what is HGPRT? Let's recall. So, it's an enzyme that is a key enzyme in your purine salvage pathway. So, when we learn about the salvage, like when you learn about the nucleotide synthesis, the unit number 3, molecular biology initial segment, where we learn about the two main pathways, the salvage pathway and the de novo pathway. Over there, this particular enzyme is learned, right? So, the T cell lack the HGPRT activity. This is the basis for using HAT medium in the hybridoma technology. B cells have it, myeloma. No worries, keep a note of this information and remember it for the further questions if by chance it is asked in the examination. And this is the importance of your previous year questions, that is, whenever there are so many questions or you will come across so many questions which you might not know initially, but during the course of learning or during the course of studying that particular question or that particular topic altogether, things become much more clear to you, right? So, make sure that you are joining up regularly. So, what is the answer for this? It is your T cells. Another question, which of the following lipid is non-ionic in nature? Yes, which of the following lipid is non-ionic in nature? Sphingomyelin, galactocereboside and lecithin or phosphatidyl inositol. Mark this. Sphingomyelin, galactocereboside, lecithin, phosphatidyl inositol. Mark the answer for this now. Very good, very good. Everyone mark the answer. And we have these classes every single day, okay? Every day I take up this session for previous year questions, same timing, 4 p.m. So, make sure that you join in regularly. Take a minute, pause and mark it. Mark this. Yes, you can try it out. But do not make a guess in the examination. I would suggest you try not having a guesswork done in the examination. Okay, right now you can do it because it's your practice session ongoing, but in the examination try avoiding guesswork. Which of the following lipids is non-ionic in nature? So it is your galactocerebroside, which is non-ionic in nature, right? So your B is the correct answer. Sphingomyelin has a phosphate group, hence ionic. Cerebrogalactoside is a glycolipid with neutral sugar head, so it is non-ionic. Lecithin, it is a Zwitter ion at pH 7, and phosphatidyl inositol is negatively charged, right? So keep a note of this particular thing. Next question, anti-B antibodies are present in the serum of which blood group? Blood group A, B, AB or O? Anti-B antibodies are present in the serum of blood group A, blood group B, blood group AB and blood group O. Mark the answer for this now. इसीलिए I tell you in the main examination it is not a correct thing to guess the answers, because guesswork backfires and guesswork should be done with what do I call, intelligence. Like you cannot guesswork out of nowhere. You need to, like, out of 4 option, if you're unsure about any 2, try to have a guesswork between those 2 and only during the practice time and not during the time when you are in your main examination hall, because over there the negative marking can lead to disqualification, right? Okay, so anti-B antibodies are present in the serum of your blood group A. ABO blood group system is there, right? Blood group A has A antigen and anti-B antibodies. Blood group B has B antigen and anti-A antibodies. Blood group AB has both antigen and no antibody. And blood group O has no antigen but both anti-A and anti-B bodies, right? it works in, no, it does not work in match the following every time also, because in match the following also correct guesswork is required. Okay, you cannot just do the guesswork out of nowhere. A smart guesswork is something that helps out. Next question, which of the following are energy requiring process? Which of the following are? So it is a multiple select question. Sodium potassium transport, non-mediated transport, active transport, and facilitated diffusion. What is the answer for this? Which of the following are energy requiring processes? Facilitated diffusion, active transport, non-mediated transport, sodium potassium transport. Mark it. Okay. Correct. Very good. Very good. So that's why I said match the following. Like whatever the question is, you have to be very smart in marking the answer. Which of the following requires energy? So B and D, where do you see facilitated diffusion requiring energy? Salil? Yes, where do you see energy required in diffusion? Facilitated diffusion does not require energy, it moves along the concentration gradient. Active transport will require it, sodium potassium pump will require it, right? Non-mediated transport is also passive, no energy is required. B and D, how can you have non-mediated transport or simple diffusion helping or using ATP altogether? Non-mediated transport also does not require it. B and D, yes. A and C are not the ones. Next question. Which of the following are correctly paired? Replication, DNA A, recombination, Rec A, DNA repair, rho factor, transcription, sigma factor. Try this. Done. Which of the following are correctly paired? Replication, DNA A, then you have recombination, Rec A, DNA repair, rho factor, and transcription, sigma factor. Mark it. One more minute, then we'll do it together. Which of the following are correctly paired? Let's see. So, see over here, replication DNA A, it's correct. Recombination Rec A mediates the strand exchange in the homologous recombination. DNA repair, rho factor terminates the transcription, not the repair. Transcription and sigma factor is also correct. So, A, B and D, all three of these are the correct ones. So, A, B and D. Again a multiple select question. Okay, the compound with higher energy, high energy compound are phosphoenol pyruvate, adenosine monophosphate, 1,3-bisphosphoglycerate, vitamin K. Which of the following compounds are high energy compounds? Multiple select question again. High energy compound is or are phosphoenol pyruvate, adenosine monophosphate, 1,3-bisphosphoglycerate and vitamin K. What should be the answer? A and C. The phosphoenol pyruvate, it is one of the highest energy molecule in glycolysis. Adenosine monophosphate is low energy. Only one phosphate bond is there, so that's not the one. 1,3-bisphosphoglycerate, 1,3-BPG, again a high energy intermediate. And vitamin K, it is a cofactor. It is not an energy compound at any point. How can you have D as a energy compound? Yes, it is used in termination. Correct. Aditi, how can you have vitamin K as an energy compound? It's a clotting factor. A and C is the answer. Let's do one more question. Last question for today. With respect to the sodium didosyl sulfate polyacrylamide gel electrophoresis, that is SDS page, which statement is or are true? Very simple. About the SDS page, it is asking you the question. Mark the answer quickly. Done? Okay. With respect to SDS page, which of the statement is or are true? Ethidium bromide is used to track the progress of electrophoretic mobility. Beta-mercaptoethanol is used to reduce the disulfide bonds. The protein migrates towards the anode, and the lower molecular weight protein migrates slower than the larger molecular weight. Mark it quickly. Last question for today. And are you people liking this segment of practicing out the questions and everything? Yes, are you people liking this concept of the previous year question paper sessions on daily basis? If yes, please make sure that you are joining on regular basis, because only then I'll be able to continue with these sessions on regular basis. Otherwise, it becomes a bit like difficult for me also to keep up the motivation and to take up the class on daily basis if you people don't join in. but you still don't join in. Salil, every day you don't join in. Okay, so which of the, with respect to SDS, which statement is or are correct? So B and C statements are the correct one. Very good. So beta mercaptoethanol reduces disulfide bonds, fully denaturing the protein. And SDS page, in the SDS page, the proteins are coated with negative charge and they migrate towards the anode. That is also correct. Gel matrix, smaller protein migrate faster, not slower. So this is wrong. And Anthedium bromide is used for your nucleic acid, not for tracking in the SDS page. So that's a false statement, right? So you can take this question as homework and we'll discuss this question tomorrow now. Okay, so you can take this particular question as homework. This is related to your hemoglobin, oxygen binding and everything. And the broken line represents the condition of that, means my graph is shifting towards the left hand side. So what will be the correct answer? This is the concept which we studied during the hemoglobin part in your biochemistry portion. Okay, so let's meet in the next class tomorrow. Be there in the class and we'll discuss the further segments. Same timing, 4 p.m. Okay, bye everyone, take care. And just a quick reminder, we have our revision capsules available. You can download our application and get to know more about it, right? So we have our GATE XL books, revision capsules, GATE XL mock sheets. In addition, rankers batch is ongoing for the CSIR examination. We have our exam cracker modules available for you all. So make sure that you join in whatever is your requirement. Okay, so bye everyone, take care, all the best.